Good morning, everybody, and welcome to the 34th Emerging Growth Conference. As always, we have an exciting list of companies and a wide range of growth sectors presenting to you today. I'm Anna Berry, and I'll be your host. So a few notes. Today, we'll be running until about 2 p.m. Eastern Time. When you switch to the next company, you're going to see a black screen for a moment, but that's just us moving over, so don't go anywhere. If you do experience downtime for more than a minute or two, refresh your browser and it should work properly again. Our platform does work best on Google Chrome, so if you're watching from an Apple device, you got to hit that play button to start the session. All of our conferences are uploaded to the Emerging Growth Conference YouTube channel, so subscribe there, youtube.com/Emerging Growth Conference. During each company's presentation, you can submit questions through our webcast module, and we'll try to address as many of these questions as possible at the end of their presentation. One last note, after today's event, you'll be redirected to the registration page for the next conference, so stay on or come back to reserve your spot early. Today, we have 6 companies presenting, so let's jump right in. So everyone, welcome Pressure Biosciences, Inc. It trades on the OTCQB under PBIO. And it's a leader in the development and sale of innovative, broadly enabling pressure-based solutions for the worldwide life sciences and other industries. You might have seen the recent press releases with some big announcements regarding their technology. So we look forward to hearing about this today. Let's welcome its president and CEO, Richard Schumacher. Welcome, Richard.
Thank you, Anna, and thanks everybody. And I hope everybody had a great 4th of July long weekend, and welcome back to work this week. Today I'm going to talk basically focused on our UltraShear technology platform, but I appreciate everybody joining us today to hear about Pressure Bio and the other great companies that will follow me. As a publicly traded company, again, I caution everybody to please do your homework before thinking about perhaps investing in our company or any of the companies that you might hear here today. We are an instrument company. We're located a little bit south of Boston, Massachusetts. All of our instruments are using very high pressure. But before I go into them, I'd like to talk about the management of the company. Myself, I'm a serial entrepreneur. I've started 4 publicly traded companies. The first 3 had a combined market cap of over $1 billion. They were Panicos Pharmaceuticals, a NASDAQ company where I was the first president and co-founder. Trinity Biotech, another NASDAQ company where I was in the founding group, and Boston Biomedica, which was my first company, BBII, which I took public back in 1996. I also spent 13 years at Harvard Medical School, initially as a scientist working in infectious diseases. I've got a great team. You can go online and hear about this team. Dr. Ting is an absolute genius. He came out of MIT with a PhD in materials management. He was a pioneer in bringing high pressure into the food industry. It's now a $15 billion market, but because of work that Dr. Ting did with Dr. Bala at Ohio State and the U.S. Army out of Natick, Massachusetts, back at around 1999, year 2000, 2005. In that era, they started using high pressure on food to extend the shelf life of food for the U.S. Army and the warfighters. But it's now grown to be, as I said, a $15 billion market. Dr. Lazarev is our chief science officer. He is an expert in the area of protein. So many of the things that we do are protein based. In fact, the things that we're doing in food and beverage now are often very protein based, as proteins are making up the viscosity and the flavor and the texture and the nutrition of the foods that and beverages that we eat and drink. Also, John Hollister joined us a couple of years ago as our head of sales and marketing. So it's a great team. The board's a great board led by Jeff Peterson. Jeff is a chemical engineer out of MIT with both a bachelor's and master's degree. Because of his background in engineering, he's been a great source of information and a great sounding board for many of the products that we've been developing here at Pressure Bio. And the other 3 members of the board, besides myself, are very, very helpful. So please go online and learn more about the board and the management team. As I mentioned, all of our equipment that we're developing is based upon the use of high pressure. Pressure is an absolutely amazing force of nature. It moves at the speed of sound. It can be controlled. It can be started and stopped. But we're the first company that I'm aware of that actually started to harness high pressure so that it can be used in the laboratory. Pressure can do many of the things that we do in the laboratory with temperature, with heat and with cold and with pH, with chemicals. But the difference is that when you use temperature and you use chemicals, when you're done your experiment, you often have to take them out or bring it back to equilibrium. With pressure, you turn it on instantaneously and you turn it off instantaneously. And we have instruments that go to pressures up to 60,000, routinely 60,000 pounds per square inch. To give you an idea, the slide here in the middle, you can see the Marianas Trench, deepest part of the ocean, 16,500, 7 miles down, 7 miles of ocean on top of the Marianas Trench. It's 16,500 psi. We go routinely to 45,000 and often to 60,000, and we do have several machines that have gone up to 100,000. It's a very safe process because we're compressing water. We're not compressing air. We take the air out of the solution. We compress the water. The materials that we want to have the effects of pressure on, the viruses, the bacteria, the cells, the DNA, RNA, protein are all in a buffer, in a liquid inside the test tube. Put the test tube in a pressure chamber like the one on the right. You can see the barocycler or pressure cycler on the right, and the chamber is a pressure chamber, and we can dial in on the computer the pressure we want to go to. So if we know we want to inactivate a virus and it inactivates or gets killed at 30,000 psi, we dial in 30,000, put the sample into the chamber, and we dial it up to 30,000 psi. Our patents, of which we have 30 patents in the area of pressure, 15 of them are in the area of what we call sample preparation. They use this instrument. We have over 350 of these instruments around the world. They're used in some of the most recognized and leading laboratories, particularly in the area of protein studies, proteomics or protein studies, for developing drugs and vaccines, for developing diagnostics, for everything from coronavirus to HIV to hepatitis B to cancer and heart disease and stroke and many other abnormalities or diseases of humans and other animals. So this system is placed in laboratories all over the world, just about in every continent of the world except Antarctica, and it's being used routinely every day with consumables. The one I'm going to talk about is this instrument on the right. This is a schematic of what we are developing. It goes up to 45,000 and soon up to 60,000 pounds per square inch. We call it ultra shear technology, or UST. We'll talk about it in just a minute. So there are three pressure technologies, platforms that we have. The first I just mentioned, pressure cycling technology, using the instrument I just showed you, the barocycler. Pressure cycling, we cycle pressure, we turn it on and off like squeezing a sponge. We squeeze cells until they break. We're able to break the cells, and the cells can be viruses, bacteria, it can be human, animal, it can be cancer, normal, etc. When I was a scientist at Harvard many years ago, we were told just beat the living heck out of the cell to open it up and get all that good stuff out. We know now that that is not a good thing to do. You want to be very careful on how you break a cell because you don't want to modify, change, destroy, alter the materials you're getting out of the cell. And so when you put them into a pressure chamber like our barocycle and you dial in pressure, you're very reproducible in how you're breaking that cell. So again, we have some of the key opinion leaders in the world in proteomics using this in discovering markers of disease, cancer, abnormalities, etc., developing vaccines, developing antibiotics, etc. But the second platform we have is called Barofold. Using pressure, we can fix problems that occur when you make proteins in the lab. Remember, a lot of protein drugs, maybe about 20 to 25% of all drugs on the market, are protein-based. They're very good because they're very specific. It's like a key and a lock. They go right to the site that you want them to go to in the body to deliver chemotherapy or to bind certain things. And but the problem is that when you make these in the laboratory, the protein, which is made up of a lot of what we call peptides that fold over each other, may be misfolded. Maybe one of them is slightly off. We can fix that by putting the protein under pressure, which actually opens up the protein. And then when we take pressure off, it refolds. And in some cases, it refolds the way Mother Nature wanted it to be. So when there's a problem with a pharmaceutical company trying to make a protein-based drug, and that problem is that the proteins are sticking to each other or they're misfolded, our BaroFold technology, in which we have 8 issued patents, can sometimes fix that problem. So we're very excited about this technology too. But the main purpose of today is to update everybody. I've been here several times, which I appreciate the invitation again from Emerging Growth and the terrific team there. We're here to talk about UltraShear technology. This is a technology that we've been working on for 5 to 6 years. This is a technology whose time has finally come. We are so excited about UltraShear technology now. This is the technology that came from the first two, learning about how to cycle pressure, learning about how to use pressure to unfold and refold proteins. We came up with a solution called UltraShear technology, funded initially by $1 million from the NIH, and then funded by the U.S. Department of Agriculture for another $1 million. We've been working on this technology platform for almost 6 years. And as Anna mentioned in the beginning, we've announced in the last month in a couple of press releases that we're ready to finally take it to market. And our first product that is made by UltraShear technology will be out in the coming months. So very excited about that. So let's figure out and find out what UltraShear technology is all about. Before I do that, I did put in one slide to give everybody an idea that we do have in 225 customers. And many of these customers that are buying our pressure cycling or using our BaroFold technology are the same customers that are going to need UltraShear technology. In fact, we're already working with some of the customers that are on this slide, already talking to them about UltraShear technology, or UST. Now, what is UST? First of all, we should talk about emulsions. You know, what is an emulsion? An emulsion is a mixture of 2 or more liquids that are not supposed to go together. They're miscible in each other. The example that everyone pretty much knows is oil and water. Remember, oil and water don't mix. We're told that from the time we're 2 or 3 years old, and we see it baking in the morning, pancakes or whatever it is, and you put your oil in the water. They just don't mix. And when they don't mix, that can be a problem when you're using an oil that contains an active ingredient and you're ingesting that, trying to get that active ingredient into the body. Vitamin D3 is an example. Coenzyme Q is an example. We don't absorb oil as human beings, and our pets don't absorb oil, and plants don't do a good job of absorbing oil. So whatever is in that oil that comes into our bodies is often excreted with very little of that active ingredient being taken out of the oil. So if it's a drug, if it's a therapeutic, if it's prednisone, for instance, a cream that we're putting on a rash, and and the prednisone active ingredient is in an oil, they tell you to put it on liberally. Or if it's another therapeutic that we're taking orally, there's a sign on the side that says mix well before using. Why? Because emulsions separate out. Most emulsions are what we call microemulsions. We mix them up just like putting oil and water. If we take a beater and beat it up really fast for a while, we'll break the oil up into tiny little bubbles, but eventually in the next 5 minutes or so, it's going to coalesce and go back together, and we're going to separate. And if that happens with a drug, of course, we may be just taking water and not the drug itself. So it's very important to mix it well before using. But even after mixing it, we're not going to get as much. The bioavailability of that active ingredient is not going to be where it needs to be. So emulsions are in our entire life. There's thousands and thousands of products in our everyday life that are emulsions. Think about our raspberry vinaigrette salad dressing. You let it sit for even 30 seconds, it separates. Milk, if you let it sit for half an hour, will separate. Many, many emulsions that are in our life. Pharmaceuticals, as I mentioned, health and wellness products, cosmetics and personal care. There's a lot of emulsions that are used in cosmetics. Agrochemicals are emulsion. Many are emulsions. Essential oils are now being used because they have a natural ability to be a pesticide or insecticide, but they'll separate out. In food and beverage, as I mentioned, milk and juices and salad dressings and many, many other mayonnaise. These are all emulsions. The problem is that Historically, most emulsions, when the food and beverage industry or the cosmetics industry or whatever is putting together their emulsion, most of these emulsions are microemulsions where they are not very soluble. The oil, which contains the active ingredient, is not very soluble in water, and therefore they remain pretty unstable. They'll separate out over time, and they will not last for a long period of time. In fact, most people tell me neem oil that they buy at the local hardware store lasts 2 or 3 months, and then it separates out, and then it doesn't work that well anymore. So the poor bioavailability is an issue with the therapeutics that are made up of emulsions. The solution is what we call nano emulsions. Nano emulsions is a buzzword that's happened, come out in the last year or two, a lot of it being driven by the cannabis market, the CBD and THC market, particularly the CBD market. So the nano emulsions are known throughout 50 years of studies on small scale nano emulsions in laboratories all over the world to be highly water soluble, to have exceptional bioavailability. The WHO sent out a report about 4 years ago saying that CBD, an active ingredient in oil, is only about 6% bioavailable, which means 94% of the CBD you're taking in a macroemulsion or microemulsion is being excreted from the body without ever being absorbed. It can't do its job if it's not absorbed by the body. But nanoemulsions have excellent bioavailability. They have excellent long-term stability. And when you know you have a nanoemulsion, the oil droplet is so small that the sample becomes actually translucent. You can see through it. When you get to the point where the oil is smaller than the wavelength of light, light goes through it and becomes translucent or transparent. And then you know you have a nanoemulsion. And if you have a nanoemulsion, then science tells us it's going to have great water solubility, great bioavailability, very long-term stability. Current processing methods for emulsions, mostly microemulsions, are not that good. Ultrasonic, which is high frequency sounds, they take a long time for the treatment. There's hot spots. There's poor reproducibility. There's sometimes cavitation, or some of the metal comes off of the sound source, the sonic source, and it's very difficult to scale if you're in manufacturing. Pressure homogenizers, like the ones that we have developed, usually have issues where the product enters the pump, and then you have to clean the pump, clean in place issues that everyone has. The valves often clog. I've talked to a number of people that have the pressure homogenizers, and they say the biggest problem is they get it going, and 5, 10, 20, 30 minutes later, the valve is clogging. They're expensive to scale. And nano results are real nano results aren't really seen that much even in these pressure homogenizers. Vendors often say they have a mystery emulsifier cocktail. Emulsifiers are these chemicals. We use all plant-based natural emulsifiers that keep the oil from reaggregating, and others use synthetic materials as their emulsifiers. We don't. Everything we use is green. It comes from plant sources. So UltraShear, this is the answer to how to make a proper nanoemulsion that is translucent, has long-term stability, has exceptional water solubility, and therefore should have exceptional bioavailability. We've been working on this for 6 years, and very, very happy to have announced twice in the last 30 days that we're teaming up with one company, and also announced that we will be teaming up with other companies in multiple areas of using UltraShear technology to make nanoemulsion in applications, not just in cannabis, but in multiple nutraceutical supplements, wellness products, in agriculture, in many other ways, pharmaceuticals down the road, of course, and in cosmetics. So there's so many areas that we can take this amazing technology that it gets us out of bed, with a smile on our face nowadays. So UST uniquely combines ultra high pressure with intense shear, and so these two make 1 + 1 = 50 in our estimation. UST produces very high stable, very long-term stable, water soluble oil and water nano emulsions. We define nano emulsions as under 200, what's called nanometers. We actually, all of our nano emulsions so far have been under 100 nanometers. So they're actually the oil that we break oil drops into millions, if not billions, of nano-sized oil droplets. So we spread that active ingredient to 1 billion different nano-sized, usually under 100 nanometers, smaller than the size of a bacterium. And we spread it out so it becomes much more available, much more absorbable by the human or the animal body. It becomes clear. And actually, there's been a lot of indication that the texture, the taste, the smell is actually opened up when you go into a nanoemulsion. Whether it's our nanoemulsion or anybody else's, if you make a good, true nanoemulsion, you actually should not only extend the shelf life, but also it should smell and taste and be as nutritious, if not more nutritious, than it was as a micro emulsion. We also believe, we have yet to prove it, but we believe, and our collaborators at Ohio State believe, that we are inactivating or destroying the pathogens. So we're actually making the product safer in the case of microorganisms. We will prove this, right or wrong. We believe it's true. But again, I caution everybody, we still have to prove it. We have already 7 issued patents, including 2 in the U.S. on our nanoemulsion technology. Let's see. This is a conventional homogenizer, not ours, and conventional homogenizer where the oil on the left is going through a fixed orifice, and what happens is that fixed orifice gets clogged with all of this debris, and then you have to shut everything down and then clean it and then get it going again. This is our dynamically controlled valve. This has a U.S. patent that was issued several months ago, as well as patents throughout the world. We have 7 issued patents, and we have more coming. And what we did is we put a back pressure to it with a ball that keeps the orifice at a specific level. But when it starts to clog, the ball backs off and the orifice opens up a little. The material flows through, and we capture it the next time around, where we shear it on the next time around or the time after that. So we don't have a clogging issue, which is a major, major problem with others that are using high pressure instruments in making their nanoemulsions. As I mentioned, it's a broad market potential. Creams, jellies, therapeutics, our coffee, our additives to coffee, agricultures. All of these have emulsions that can be made into nanoemulsions to make them much better. I'm going to show a few slides on some data. I'm not going to go into any detail. I'm happy to take phone calls anytime after this. The questions we'll have, and then I'm happy for the rest of the week and next week, whenever, to take any questions. This is on prednisone. Many of us at this talk today have taken prednisone, most likely for skin rashes. But again, you need to get through the skin. It needs to be a topical that can be absorbed through the skin in order for it to have its best efficacy. So here's prednisone. There's always two different lines on this. The first one is talking about the size of the particle, of the oil particle. You can see here that the size of it is going down to, and then across the board is the number of cycles. So as we increase the number of cycles, as the material goes through our machine, 1 cycle, then again a second cycle, then again a third cycle, as each cycle happens, we're reducing the size of the oil drop into very small nano droplets. And you can see it goes from 200 nanometers down to 100, down to 50, down to 40, down to 35 nanometers. But more importantly, or as important, as it's going down, as we're increasing the number of cycles, we're also making it more transparent. We're giving it more clarity. So when you're putting a nanoemulsion into water, into a drink, you don't want to see gunk floating around or discolored floating around. You don't see that with ours. We go so small that it becomes water soluble and dissipates into the water. so that it becomes very clear as you're holding it. It's very clear. So this is prednisone, and it shows the lower that we go, the number of cycles as we increase, we decrease the size of the oil droplet until it becomes a nanoemulsion, which we consider under 100 nanometers, and it becomes very transparent. Here is retinol. Retinol is a compound that's used in a lot of cosmetics. Again, as we increase the number of cycles, the size of the oil droplet, which was rather large to begin, goes down to where it is around 54 nanometers. But as we increase the number of cycles to get down to very small droplet sizes, the clarity goes up, so it becomes much more clear. Same thing here with neem oil. Neem oil is a natural product that has a natural pesticide ability for ants and other insects. And as we increase the number of cycles, we decrease the size of the neem oil droplets, and we increase the clarity of the solution. Now, at the same time, we're increasing the stability, and if it's a food or beverage, we're also increasing its shelf life. And in some cases, increasing its opening up its flavor and keeping its nutrition. Astaxanthin is considered by many the world's most powerful antioxidant, and it is coming out of krill that are found in the ocean, and out of algae in the ocean. And you can see here we've got a picture of the initial product that started at about 10,000 nanometers, and we got it down to 47 nanometers. We're working with several companies that are astaxanthin suppliers in the world. They're quite honestly, I think they're astonished at what we can do. So you can see the beginning part of astaxanthin, very thick red solution. And as we go through 10 or so cycles, it becomes very, very clear, much more. And this is something that you take as a supplement. And therefore, I mean, this is a lot more appetizing than this blood red initial sample. And you can see the oil droplets here pre-UST and after UST in a microscope. You don't see anything because they're under 50 nanometers, so you don't even see them in a regular optical microscope. This is krill oil nanoemulsions, which is high in omega-3. A lot of us take omega-3 as a supplement. Again, omega-3, we can turn it into a nanoemulsion. So we've brought it down to about 100 nanometers. This is a tough one, but we've been able to go from almost 220 down to about 100 nanometers. And then as we've done that, we've made it very clear and much more water soluble and much more bioavailable. This is algae oil nanoemulsion, same thing. As we do cycles in the machine, each cycle takes just a couple of minutes. We can decide where we want to go. We can actually dial into our machine the nano. If we want to get to 100 nanometers, because that's good enough, then we can stop it after 1 or 2 cycles. And we want to get down to 50 or even below 50. Maybe we go 4 or 5 cycles, but we can determine this. It's very reproducible. This is curcumin. As an anti-inflammatory. A number of people will take curcumin as a supplement. We've taken curcumin down to 37 nanometers and made it extremely clear. We have taken, sorry about that, thought that was off. We've taken CBD, which is what we consider the low-hanging fruit with CBD. CBD is a molecule that comes out of the cannabis plant or the hemp plant. And so it comes out in an oil. So when you're taking CBD, you're taking a CBD oil if you're taking natural CBD. And again, it's not going to be absorbed very well. We've taken CBD oil, nanoemulsified it down to about 20 or 30 nanometers, and then we've got a very clear solution. This makes the CBD oil far more absorbable. So there's a lot of people out there that think that the CBD that a lot of people are taking right now is really not being absorbed that well because there aren't many nanoemulsions that are being used in CBD, and if they are, the nanoemulsions themselves may not be as high quality as we believe ours is, because we've tested a number of them. This is conventional process CBD oil. This is what is put into a lot of gummies and put into some drinks, etc. And this is the same material that was processed by ultra shear. You can see how clear it's become because the oil here was broken into a tiny little, billions of nano-size oil droplets, all containing CBD. The CBD still works as well as it did when it was here, except when you take it, it's far more absorbable. You can go from maybe 10% bioavailability to 90 to 100% bioavailability. The clarity again shows that you've made a nano emulsion. This is the same material that 730 days later. So we have over 2 years of stability now in our CBD, and it looks as clear as it did on day one after we first processed. Very, very happy. We haven't seen anything like this from any other manufacturer of CBD that's been published. This is the best we've ever seen and the best that people we talk to have ever seen. Here is a serial run of CBD as we decrease the size from of the oil droplet from 140 down to close to 20. We increase the clarity. You can see the oil floating on top of the water here. And then as we do 1 pass, 2 pass, 3, 4, 5 here, all the way up to 6 passes, you can see that the oil is now being broken up into tiny nano-sized particles, so it goes into the water. So here, after 6 cycles, we've hit the limit here, 47, 46 nanometers, and you can see how clear it is. It will stay this clear, we believe, for the next 2-3 years plus. So the bonus is, as I mentioned, that we believe that we're killing the bacteria and viruses, but if we're not, because our nanoemulsions are smaller than bacteria, we're able to filter sterile. So when we're done with making our nanoemulsion, we put everything through what's called a 0.2 micron filter. So if anything did get through, it's going to be filtered out. So the material that goes through the filter is completely sterile nano emulsion. And that's what we're coming out with in the coming weeks. This is a store-bought so-called nano emulsion. We've done this a number of times. We've gone out and purchased other people's nano emulsion, and almost invariably when you put it into water, this is what you get. You've seen that earlier. When you put ours into water, it dissipates into water. It's essentially water soluble. It remains clear. So this could be put into spring drinking water and you'd never know it was there, and it'd be very, very bioavailable. So the last couple of slides, recent news, I'll start at the bottom. We sent our biggest machine, our first commercial machine, to Ohio State. We have a collaboration with Ohio State. We have a consortium that we're each 50% leaders on. This is a consortium for introducing UltraShear to the food and beverage market around the world. Ohio State's got one of the top colleges of food, agriculture, and environmental science in the world. We work with Professor Bala there, and they are now becoming experts at using our instrument, and they are inviting a who's who list of food and beverage companies to come in and look at UltraShear. In April, we announced very strong results for year to year for 2021 versus 2020. We announced in April an early access program, and this is where we found Safer Medical of Montana, the first company that we've signed a deal with to distribute a product. Our first manufacturing agreement under the early access program was signed and announced on May 4th. And then on June 29th, we mentioned that the first ever UST processed nano-emulsified CBD product, it's going to be an aerosol spray for oral use, this is the first product under this agreement. And Safer Medical expects to have this out on the market by no later than around Labor Day, and to sell this material through the end of the year. And over the first full year, Safer Medical thinks that they will do about $3 million in sales. This is a slide about Safer Medical of Montana itself. Our partner, Captain Ackerson, the founder, served 2 terms of duty in the Army. The first was when he was 19 years old, and he was a helicopter pilot in Vietnam. They'll be distributing this product, which is a oral aerosol for oral use. The formulation was completed. It took about a year because we met them initially about a year ago. First pilot production run was run. They sent it to some of their sources, and they got back what they call better than expected results. We're going to go to market in Q3. He's going to sell the product through his website and his already existing distribution channels. And he's also setting up groups, channels with veterans groups such as nurses, 10,000 nurses that work with veterans around the country. Retail sales will begin in after Labor Day, and the first lot is going to be around 40,000 vials. Expected price around $63 a vial. They're targeting about $3 million over the first 12 months of selling this product. We've got a deal with them with a very good revenue split for Pressure Bio. We're getting about a third of the revenue, so we expect about $1 million over the first year of sales of this product. We expect this product will sell for far more than 1 year, and that the volume itself will go up. In fact, Safer Medical believes that eventually they can be doing 500,000 vials. You can do the math. That's an amazing number. They are donating all their profits to different veteran groups. As Captain Ackerson said, he's had a good life. He's done a lot. He wants to give back. And I will say that the Safer Medical transaction, we believe, is going to be the first of many similar ones of UST processed nutraceuticals, cosmetics, and many other areas. So I would like to thank everybody and open it up now for questions, Anna.
Thank you for this great presentation. All right, let's jump in. Dan Morris says, you mentioned that PBI could potentially bring in over $1 million in just 1 year from just one contract. Is that a representative contract for more to come, or is this a contract exceedingly aggressive? And will future contracts be for significantly less revenue?
We don't believe future contracts will be for less. We actually think future contracts will be for more revenue. I was asked when I gave a talk last week about what's so important about this first one. The world has not seen, we believe, anything like ultra shear technology processed material, nano emulsions. These are very, very, very high quality. As an example, we gave one of the samples went to one of our investors who sent it to his son, who's an athlete in college, who immediately said to his father, "This is the best looking nanoemulsion spray I've ever seen." And he called him a week later, as I understand, and said, "Dad, this thing works. I'm going to invest in the company. This thing works great." It's a true nanoemulsion. And so we believe that the first one is always the toughest. We got to get a name for ourselves. But once the public, so once the consumer sees how well this works, we believe that the following, that will sell more through Safer Medical than the 40,000 vials we're initially doing. But we also believe that other contracts are coming and coming soon for other products, and they're all going to have the same high quality. And that once people see how good a nanoemulsion by UST is, it will get a name on its own, and it will sell far more than we're going to be selling in the beginning. That's our belief.
Great. Carl C. says, if PBI can average 1 million per contract per month, that would equate to over 12 million per year. Is it possible for PBI to do that many contracts in a year, like in 2023, for example?
Well, not in 2022, Anna, but I'm glad you said 2023, because we do believe that $1 million per contract is a doable number. And we do believe that we can sign 12 contracts next year. And so, yes, that's a doable number. We're not giving, I'm simply saying it's doable. I'm not saying we're going to do it. I'm not giving any future looking revenue advice, but I am saying that, yes, if the question is, do we think that $1 million a month, $1 million a contract is a fair number? The answer is yes. And do we think we could sign 12 contracts next year? The answer is yes, we believe we could.
Great. Tanya Curry says nano emulsion is a new buzzword in supplements, vitamins, nutraceuticals, as well as in markets with other concentrations of the oil-based products like agrochem, cosmetics, and pharmaceuticals, for example, like you mentioned. The question is why? What do they do that the microemulsions that have been used to date do not do?
Yeah. So as I mentioned earlier, there's a lot of pretenders out there, unfortunately. There are in any industry. There's a lot of pretenders. There's a lot of groups out there that are saying they have nano. Certainly there are some that truly have nano. Can they do it as efficiently as we can at a lower price cost and at a higher quality that we have? I'm not sure. But there are some companies that certainly have nanotechnology. But there are many out there that we've tested that call it nano, and they're not nano. So if it's not nano, it is not going to be absorbed that well. It's not going to be that water soluble. It's not going to be that bioavailable. It is not going to have that longer shelf life. So I've told my guys, I know Intel is probably the best marketing job I ever saw in my life, Intel Inside. I think we should put UST processed on everything that UST is processing, because if it's UST processed, you can be assured that you're getting the highest quality nano emulsion of that product that you can get today in the world.
And Paul Hunter asks, will PBIO be responsible for 100% of the manufacturing of this new nano emulsified CBD oral product?
That's a good question, Paul. No, we are nano emulsifying it and developing a stock solution of nano emulsified, in this case, high quality CBD nano emulsified by UST. We will then supply this to Smarter, Safer Medical of Montana, and Safer Medical will then send it to their bottler and vialer, and they'll have it bottled and vialed and ship it out under their name. We're not a company that's going to have ready-to-use products being sold directly to consumers. We're going to sell the product in tandem, in partnership with other companies that are already selling products to consumers. And but we're setting it up in the beginning to share. We have something that we don't think anybody else has in the world that can compete with us in the world, and we're giving that to distributors. So in return, we like to share in the profits. And so far, we're talking to at least a half a dozen companies in earnest, and these companies are all agreeing so far to work with us in sharing the profits.
Great. Last question due to time. Arnold Santos wants to know, $2 million in funding for Ultra Shear. Were these grants? If not, what are the terms of repayment?
Yeah, they were grants. There's no repayment. The government's been great to us through the pandemic and prior to the pandemic. So we have certain things we have to meet, and we've met them. And Ohio State has certain things to meet, and they're meeting them. But these were grants. They do not have to be paid back.
All right, Richard, fascinating stuff you've got going on over there. Thank you so much for joining us again, and congratulations on these updates. We have a lot more questions for you, but we'll send them to you so you can answer on your own time and come back and join us again.
I'll do that. Thank you.
Okay, everyone, stay with us. We're going to transition to our next presenter, but we'll be right back.